Move on to our second speaker, who is Parag Juvale. He is a graduate student in Dr. Sarah Pallas's lab at UMass Amherst. Their research involves understanding how sensory experience influences the development of GABAergic signaling and regulates neuroplasticity, topic that's close to my heart. So I'm excited for this. The title of this talk is Maintenance of Refined Visual Receptive Fields through BDNF-TrkB Signaling Depends on Presynaptic GABA levels, not postsynaptic GABAA receptors or the scaffolding proteins gephyrin and PSD95. Welcome.
Thank you. So that was a big title, so we changed that a little bit. But thanks for the introduction and thank you for being here, everyone. I will share my slides now. So I'll talk about my work in the Pallas lab, looking at how neural plasticity shapes the visual system development based on early visual experience. But before that, I would like to just go over a few concepts depending how the brain matures is something that neuroscientists have been trying to understand for a long period. And critical periods have been found in developing organisms where the organism learns faster during the earlier part of their life compared to the later part of their life. This is primarily because during this certain period when the organism learns faster, what happens is there's heightened neural plasticity, and that coincides with that learning, the experience part of their life, and that helps them learn something better. Say an attribute like learning a language or something, you learn better as a kid or during childhood compared to adulthood. So this is because there's this neural networks that are formed because of, I think we all here know, excitatory and inhibitory neurons. Inhibitory neurons shown here in green and excitatory shown here in blue. And this region is where the synaptic part where all the interactions happen. Just a quick refresher of synapses. Excitatory synapses, the excitatory neurons, which are shown in blue, they release glutamate onto NMD or glutamate receptors, allowing the channels to open and sodium influx into the postsynaptic cell, leading to firing of that neuron. And inhibitory neurons, like the name suggests, stop all of this action by releasing GABA onto GABA receptors and leading to chlorodyne influx into the postsynaptic cell. We are interested in inhibitory synapses. So these networks, these highly plastic or constantly changing networks, when they move through this critical period and when they're provided with that visual experience, those networks consolidate over a long time. They start forming stable networks, and biochemically and structurally, they are consolidated to form those networks for a long time. And this is important also in rodents because, like humans, the vision is initially blurry, but as the animal matures, that vision becomes clearer. And what happens behind this, what's happening in the brain or the network connections is that the receptive field size reduces. So when the animal has blurry vision in the initial few days of life, receptive fields are higher or larger. And as the animal matures, the receptive fields become smaller. So receptive field is basically the representation of the scope of vision or the field of vision onto neurons. So in this case, a single neuron is taking up information from this much area, let's say, of the scope of the field. And as the animal matures, that area reduces. This happens because of a lot of reduction in excitatory signaling, or basically increase in inhibition or GABAergic signaling. So basically, to summarize, if there's more GABA signaling, there's reduction in plasticity, smaller RF sizes or receptive field sizes, clearer vision, as against if there's less GABA, then there's higher plasticity, enlarged receptive field sizes, and clear blurry vision. So what was seen in dark-red animals was that initially the animals had blurry vision and obviously larger enlarged RF sizes, which slowly became clearer. But then during adulthood, these animals went back to their enlarged receptive field sizes, and the vision became blurry again, which means these animals had plasticity in their adulthood, which is a little intriguing and surprising because that's not what normally happens. So what our lab showed in the past decade or so is that if you give visual experience, this is something that was known. if you give visual experience during the critical period, the receptive field sizes reduce around P60 and they remain like that throughout the animal's life. Whereas if you do not give that light experience during critical period, the development of that visual system happens normally. However, that is not maintained. So what our lab showed is that visual experience is not necessary for the refinement of receptive field sizes, but it is rather necessary for their maintenance, which is a really interesting finding, because it was normally thought that visual experience would be necessary for development of the visual system. So we also showed that in the visual cortices and superior colliculi, which are associated with visual processing in the brain. And later on, we moved on to different time points or different periods. So we have showed that if light was given only during P33 to 40, then the receptive field sizes remain smaller, which means the vision was clear. Obviously, these are the basic two instances where with light, the receptive field sizes are smaller. With if the animal has been raised in the dark, they're darker. And then P33 to 40, where they again become smaller, even though they are in the darkness throughout their life. If light was provided around P60, they still were able to maintain their receptive field sizes, which means their vision was good. But if you provide light at P90, then the receptive field size is enlarged, which means they cannot see clearly. More recently, what we've shown is TrkB activation also acts as, does the same function as light, because merely activating TrkB allows the receptive fields to be smaller, even in animals raised in darkness completely. Similarly, when we provide TrkB antagonist along with strobe light, which has the similar properties of having a visual experience during critical period, the receptive fields still become enlarged, which means TrkB, we recently showed that TrkB is necessary and sufficient for maintenance of visual acuity. And the reason is because TrkB is the receptor for BDNF, brain-derived neurotropic factor, and that binding of BDNF allows for phosphorylation of TrkB, leading to PLC gamma activation. PLC gamma cleaves PIP3 into DHA and IP3. IP3 goes into the smooth endoplasmic reticulum and allows calcium to be released into the cytoplasm. Excess cytoplasmic calcium and DHA can activate PKC, and PKC leads to stabilization of GABA receptors in the synaptic region, which are held together by the scaffold protein gephyrin, or gephyrin, however people pronounce it. So this is the idea behind what we are doing for now, or what I'm trying to look at now. And just an idea about the GABAA receptor. It's a pentameric receptor with 2 alphas, 2 betas, and 1 gamma subunit. The prominent receptors in our context are alpha 1, which is GABA receptor at mature state, alpha 2, which is GABA receptor in the immature state, and alpha 5, which is an extrasynaptic GABA receptor subunit. So what we saw with the visual experience, that there is receptive field maintenance. Other researchers have also shown that initially, in the first few days after birth, the prominent receptor subunit is alpha 2, and around P12, so somewhere around here, that switches to alpha 1. So based on this, what we hypothesized is that if there's RF plasticity, then probably during P60, alpha 1 might be switching back to alpha 2. The other thing that could happen is that alpha 1, there's increased alpha 5 at the synapse, which is an extrasynaptic receptor, leading to changes in tonic and phasic inhibition. And that might be affecting GABAergic signaling in the synapse, leading to receptive field plasticity. So for this work, we use light-red and dark-red hamsters. Light-red hamsters see 12-hour day, 12-hour light cycles, and dark-red hamsters never see any light. We take their brains, make protein lysates, and run Westerns all day long. It doesn't help. But this is what we found was that there is no change in alpha 1 to alpha 2 receptors ratio, or alpha 1 to alpha 5 receptor ratio in the synaptic region, because we are looking at synaptosomes. So this is only in the context of synapses. We do not see any significant change in light-haired against dark-haired animals. We also looked at cytosol to memory ratios to see if there was any change in the rate of endocytosis between alpha-1 and alpha-5 subunits. And we did not see any significant changes in light-haired against dark-haired animals. So since we did not find anything in the prominent receptors, or the more prominent ones in this case, we moved on to the next logical step, which is gephyrin. Maybe gephyrin is not aggregating enough for them to be held at the right place in the right structure. We also looked at PSD95. Unfortunately, we did not see any changes in that either. So this is still confusing or intriguing for me because there's no change in gephyrin or PSD95. So since there's nothing moving around here, we moved on to the next part, which would be probably chloride channels, because if there's an imbalance in the immature to mature chloride channels, then probably the postsynaptic neuron is always, yes, I'm about to close up here. So that might be a problem that the ion pumps might not be functioning well. We also did not find any change in KCC2 to NKCC1 ratio or their absolute amounts in light-haired versus dark-haired. Just to be sure there's no issue with the excitation, we also looked at NR2A and NR2B, which are the immature and mature subunits for excitation or NMDA receptors. And again, we did not find any significant changes. So this is where the story is at right now. So in conclusion, we found the previous work has shown that there is a reduction in GABA. We have seen this by IHC, and GABA receptor function, we have seen that pharmacologically. But we do not see any changes in the rate of endocytosis or the type or amounts of GABA receptor, mature GABA receptors at synapse. We do not see any changes in the scaffold proteins, nor in the chloride pumps, chloride transporter pumps, and also no changes in the NMDA receptors. So for future work or the remaining part of my PhD, what I'm planning on doing is to look at alpha-4 subunit because it has been implicated in Fragile X and epilepsy. And alpha-4 is also an extrasynaptic receptor subunit, which can move into the synapse and create all these problems with inhibitory network. The other thing is to try and understand why TrkB activation is sufficient to stabilize the inhibitory network. For that, I'm planning on mass spec so that I can get more. Maybe there's nothing wrong with the actual receptor subunits, but there could be adapter proteins or excess phosphorylation, which is leading to this problem. The third thing, which I find really interesting, is to understand how visual experience at P60 is equally efficient as early visual experience. So one idea, one hypothesis I'm having here is that probably mRNA, which can generally stay up to 2 to 3 weeks after the activity, might be present in the synaptic region, and that is leading to a lot of proteins for the next 2 to 3 weeks. So beyond P40, 2 to 3 weeks would be P60. So I'm assuming that could be one part. So we're also considering doing something like RT-PCRs or RNA-seq to just understand what's happening at the synapses. With that, I'd like to thank my advisor, Dr. Sarpas, members and past members whose work I've shown today. And
thank you. That was lovely. I do have one question, and that I do have a few questions, but the first one I have is, I know nothing about the hamster visual system. So how does that compare to, for example, a mouse or ferret, where most of these receptor fields and early critical period work was done with?
Yeah, so actually that's interesting that you say that, because even I'm having problems locating hamsters because no one works here on hamsters, and COVID has changed everything. So for 3 control hamsters, I have to wait for like 8-9 months now. But the visual system, the development of visual system in hamsters happens a little later as compared to mice and rats. So we also work, one of the grants that we have is on looking at 5 different animal models, because the idea is that ecological niches change how animals perceive things and see things. For example, we were also working on ferrets, where they're predatory, their vision needs to be even better, whereas compared to rats and mice, they hear something, they see something, and they start running. So, but in hamsters, to answer your question, hamsters have a visual system that have development that happens post-birth. So that helps looking at all of these things. because my lab primarily works on electrophysiology and behavioral changes. So it helps them to see those things.
One other question I have before we move on is, have you considered looking at immunostaining type approaches, mainly because, right, the composition of the GABAA alpha-1 receptor versus alpha-5, there seems to be some indication that they are on excitatory versus albumin positive GABAergic neurons, right? The alpha-1 being a gating thing mostly in albumin versus the excitatory. So I was curious if you've.
That's actually interesting because two things. One is that is small caveat of this work because we don't know which inhibitory neurons we are targeting because we're just crushing everything. About immunosuppression, we have tried it to some extent, but alpha-1, this is I've done Westerns all my life. This is the worst thing I'm doing. Alpha-1 is the worst. Yes, seriously. I mean, you cannot get good antibodies. Alpha-1, alpha-2, alpha-5, they're almost the same. There's minor changes and amino acids. It's super hard to get those immunostaining done. It would be part of what I'm planning on IHCs after this. But GABAs and PSD95 and all of those we could stain easily. Alpha receptors, it's a problem. That's why my plan is to look at mass spec, and that might give more amino acid based changes. So probably we might have better answers there.
Right. Just as a quick aside, so Fritschy had a protocol. You had one of his references up there. It's the Nature's protocol from 1998 or something like that, where they do GABA-A alpha-1 receptor subunit Nature protocol, which works beautifully in mice. So I've done that before. So
I will email you, obviously. Now that I know you're also interested in this thing. But yeah, I will.
Well, thank you again.
Yeah, I really need some good data at this